All right, good morning, everyone. Welcome to our 11 o'clock briefing. The viral microbiome gets a leading role. We have three terrific speakers with us this morning. They are Joshua Weitz, Professor of Biological Sciences and Director of Quantitative Biosciences, doctoral program at the Georgia Institute of Technology. Matthew Sullivan, Professor of Microbiology at Ohio State University, and Alison Buchan, Professor of Microbiology at the University of Tennessee. And we'll get started this morning.
Great. Thank you for joining us today in this briefing highlighting new and ongoing discoveries of how viruses reshape the fate of cells, populations, and even ecosystems. As you know, viruses need a host to replicate and survive. It is tempting to imagine that the most common host is us, humans. Indeed, viruses that infect humans, as you see here, like influenza, Ebola Zaire, HIV, or Zika, can have devastating and often lethal consequences. Yet most viruses on Earth don't infect humans. Instead, they infect microbes, including single-celled bacteria and archaea. That is to say, microbes get sick too. Viruses that infect bacteria are known as bacteriophage, where the term phage comes from the Greek root phagos, meaning to devour. These viruses are in many ways bacteria eaters, but they don't literally eat. They infect, replicate, and spread with a life cycle of their own. To understand the life cycle, consider a virus particle, 50 nanometers in size, which is about 1,000 times smaller than the width of a human hair. It diffuses inside our gut or in the surface ocean. By chance, this virus particle will come in contact with the bacterial cell, and the virus particle will start to transform. The genetic material of this virus, this phage, is injected into the host cell and then redirects the cell's machinery to make more virus genomes and proteins which self-assemble into virus particles, leading to the lysis and death of the host cell, and eventually the release of new virus particles back into the environment. This process takes place in part because phage are incredibly abundant. There are more than 10 billion virus-like particles in a single liter of surface seawater. They're also highly diverse, infecting both rare and abundant microbes. For example, phage infections of globally ubiquitous marine autotrophs at the very base of the marine food web redirect carbon from those cells back into the microbial loop rather than upwards through the food chain, reshaping global carbon cycles. Yet do phage in the environment do more than kill or prepare to kill? Today I'm joined by Professor Matt Sullivan from the Ohio State University and Professor Alison Buchan from the University of Tennessee, Knoxville. Together we work on models, experiments, and field-based studies that are revealing surprising new principles by which phage form long-term associations with their microbial hosts that don't necessarily culminate in immediate lysis. The basis for these associations is the realization that rather than kill, phage can integrate their genomes into the genome of bacterial hosts. This integration is called lysogeny, or latency. Although the molecular biology of latency has been studied for many decades, the evolutionary rationale has remained poorly understood. What advantage does latency offer? Would it not be better and in viruses' interest to kill their hosts and produce hundreds, if not thousands, of viral progeny? What benefit is there to the virus to form a semi-quiescent infection? In a sequence of papers available at Nature Communications, Journal of Theoretical Biology, and the bioRxiv preprint server, and supported by the National Science Foundation and the Simons Foundation, my group has led efforts to establish a new theoretical framework to investigate the ecology and evolution of latent infections for viruses that infect microbes. Critically, we propose to measure viral fitness not in terms of particles, but in terms of infected microbial cells. This change in perspective enables direct comparisons of the benefits of latent and lytic strategies. In doing so, we measure the fitness benefits of latency in terms of the vertical transmission of viral genomes from mother to daughter cell, and the fitness benefits of lysis in terms of the horizontal transmission of viral genomes to other cells. These fitness values depend on ecological contexts. For example, we predict that lytic killing is favored in environments where there are many susceptible hosts, and latency is favored where there are fewer hosts and when virus infections directly benefit cell reproduction and/or survival. This last finding suggests how entanglement of virus and microbial cell fates can change evolutionary incentives towards beneficial viral infections. The predictions derived from this framework go beyond the predator-prey paradigm of virus microbe dynamics that has predominated in the field for over 40 years. In doing so, our group's findings consider viral fitness from a cell-centric lens, while accounting for the ways in which viruses persist both inside and outside cells. Critically, this new approach also has direct application to experiments and environmental studies of viral ecology, as we'll hear next. Matt?
Okay, we'll have the next slides. So I'm here to emphasize the powerful role of viruses in nature, particularly in the oceans. Can we get the next slide? That was Joshua's talk. Particularly in the oceans, where they're best studied. Sampling in the oceans is always an adventure. And you can see here in the video, which will crop up any minute, just how much of an adventure it can be. I'm a member of the Tara Oceans Consortium, which is a large international consortium of researchers that have studied viruses to fish larvae globally. We've now covered much of the temperate and tropical oceans. We've completed a circumpolar navigation of the Arctic. And we've recently had a focused expedition to study 3 particular coral species and the holobionts and overlying water throughout the South Pacific. I'll discuss today virus-related stories that emerge from these original Tara Oceans expeditions to focus better on understanding how microbes help the oceans mitigate climate change. The work has been published in a series of Science, 3 Nature, a Nature Biotechnology, and a Cell paper, and it's been funded by the NSF, the Joint Genome Institute, and the Moore Foundation. This is a map of carbon export in the oceans, where red is high export and blue is low export. Many of you are probably aware that we are now in unprecedented times in Earth's history where humans have accelerated greenhouse gas production into the atmosphere by burning fossil fuels and forests. However, fortunately for us, the oceans are a great sink and absorb half of this carbon dioxide from the atmosphere. Unfortunately, it comes at a major cost for the oceans: acidification and warming. The acidification can harm corals and fisheries. It can increase dead zones, red tides, and cause severe ocean-modulated weather, such as hurricanes. The fate of this ocean-absorbed carbon dioxide is controlled by ocean microbes. And a first goal of Tara Oceans was to develop planetary-scale biodiversity maps, again, from viruses to fish larvae, and use these in conjunction with big data approaches to understand which organisms drive carbon export in the oceans. In this map, we asked that question: which organisms are responsible for the red, high carbon export zones? And the Tara Oceans had also surveyed eukaryotes, bacteria, and archaea. It was viruses that best predicted carbon flux to the deep sea. And this in silico prediction from global scale modeling led to follow-on experimental work, which has turned these paradigms of viral mediated rapid recycling on its head. We thought that viruses should reduce carbon flux to the oceans, and we're now learning that carbon flux is driven in a large part by viruses. Given these ecosystem scale impacts, it becomes imperative to understand fundamental ecological patterns and drivers for ocean viruses. And by developing and analyzing a systematically sampled and processed global ocean virome dataset, we've recently learned a lot. First, in this plot, you can see that there are 5 ecological zones that emerge from the global dataset. These provide a baseline for understanding viral meta-community structure and connectedness across the global series of viral communities studied. Second, we established biodiversity hotspots, which included high diversity in the low latitudes, as we expected, but also a surprise in that there was high diversity in the Arctic. The microdiversity is the first time we're looking at this. This is looking at variation within groups that's a finer scale level than had been observed in previous studies. This microdiversity is quite low in the oceans as compared to lakes. Fourth, we evaluated these macro and microdiversity patterns against classic ecological gradients in latitude, which I've shown here, as well as depth, which I've not shown. Again, the increased diversity in the Arctic was quite a surprise and has led to a number of mechanisms that we're thinking about now in the Arctic, which will help both understand important mechanisms that are happening there, and help us establish ecological roles in the understudied and climate-critical Arctic Ocean waters. And overall, these efforts provide foundational ecological patterns and drivers, and while some of these findings support existing paradigms for microbes and macroorganisms, some suggest that viruses play by different rules. So finally, in this last slide, I want to zoom in from the global perspective to the microscale, a particular model system, a model system that we studied in the laboratory. We're using these large-scale surveys to actually tell us which viruses are worth studying, which viruses are abundant and therefore interesting to look at a more mechanistic level. From these surveys, the most abundant viruses are T4-like viruses, whose sewage relatives have been studied for decades and largely established foundations in molecular biology. However, many of these abundant viruses are not known in culture and had never been seen before. Fortunately, my group had been studying the third most abundant virus, the 538.1 viruses, which I'm sure none of you have heard of, but we happen to have been studying anyway, using a nature's mutant approach to try to understand how these viruses might behave differently in natural settings. In the current paradigm, from abrupt disruptions from genetic manipulations, suggests that host cells become resistant to viruses by single mutations. To evaluate this in nature, we studied 2 naturally occurring cells that were identical at a measurement locus. But when they were infected by this particular virus, the infection was either efficient on one host or inefficient on the other host. And using multi-omics approaches, this revealed that these nature's mutants were inefficient at many steps in infection. First, here at adsorption. Second, failing to block defenses. And third and fourth, failing to make and degrade relevant constituents of the viral particles. So this suggests that the paradigm of single mutation resistance is less relevant to natural systems, likely because of the mixtures of viral pressures that cells encounter in nature. Better understanding of this ecology of phage resistance is critical for understanding how ecosystems are affected by viruses, and also more closer to home in a clinic to try to how phage therapy treatments can be better used to defeat antibiotic-resistant pathogens. Thank you.
I'm going to go ahead and get started while they're being pulled up. So my lab studies the ecology and physiology of a specific group of marine bacteria. They're called the Rosibacters. And so Rosibacters are major players in the world's oceans. They're found in virtually every marine environment one looks, from coastal to open oceans, sea ice to sea floor, and in close association with a myriad of marine eukaryotes, including things like corals, sponges, seagrasses, and phytoplankton. Not only are Rosibacters widely distributed, they're abundant. Members can comprise upwards of 20% of coastal bacterial communities and more like 10% of open ocean communities. Furthermore, these bacteria are a metabolic tour de force, making critical contributions to marine carbon, nitrogen, and sulfur cycles. For these reasons, Rosibacters are environmentally relevant models, and fortunately for us, representatives can be readily cultured in the lab for study. So a common feature amongst cultured Rosibacters is the presence of temperate viruses, or phage, in their genomes. These are the types of viruses that Joshua mentioned earlier. They can integrate into the genome of their host bacterium, often at a site-specific location, where they can replicate along with their host in a non-lethal manner. Or they can initiate a lytic mode, where they excise out of their host genome, reproduce in mass, and lyse that individual cell. So viruses of this type enter into really long-term relationships with their hosts, some of which can be mutually beneficial. What I want to share with you today is the story of a Rosibacter strain we isolated about a decade ago, along with its 2 infecting viruses. On this slide, I'm using cartoons to indicate the players. We have a single bacterium that is infected by either one of 2 viruses. They're indicated as either red or blue within the genomes of these little bacteria. When we look at the DNA sequences of these viruses, we see that they're nearly identical. Furthermore, they share an integration site in their host. Of particular interest, we've found that which virus the bacterium is hosting influences the physiology of that bacterium. An example is shown in the bottom of the slide in the difference in the way that these 2 bacteria-virus partners are able to colonize a surface and grow as a biofilm. In this instance, when the bacterium is harboring the red virus, it's a much better colonizer than when it's harboring the blue virus. Perhaps what's even more intriguing is the way that these two virus-bacterium pairs interact with one another. As I mentioned earlier, these genome-integrated viruses can sometimes turn the tables on their host cell. When and why they might do this is a subject of current debate. Regardless, when this happens, the virus makes many more copies of its genome, packaging it up into protein, and then bursting free of its host cell, killing that individual cell in the process. These progeny viruses set out to encounter a new susceptible host. In the instance that's indicated here in this video, a cell harboring the blue variant of this virus is susceptible to infection by a red viral particle. And please note we took a little artistic license here. These viruses wouldn't be preformed in the cells. But one outcome of this interaction is a lytic infection of the blue cells when attacked by the red viruses. And during this, red viruses are made and released from cells. However, during the course of the infection, the blue virus is prompted into action, making many more blue viruses that can then turn around and attack the red virus harboring cells, resulting in a similar outcome, the production of both blue and red viruses, setting up a competitive interaction between the two host virus pairs. So whether or not this type of interaction occurs in nature remains to be seen. But if it does, it may prove to be a newly recognized mechanism for stabilizing the population structures of ocean microbes. Thank you.
Thank you. Okay, so we'll take questions from reporters. If you have a question, kindly raise your hand. We'll bring a mic to you, and please state your name and affiliation before your question. Okay. And maybe while we were waiting for our first question, I think. Oh, do we have one here? Okay, front row. Here's the mic.
Hi, I'm Kathleen O'Neill, freelance. For Dr. Sullivan, you had mentioned that the viruses, you're looking at the distribution, the microdiversity and the macrodiversity. Can you explain when you said viruses play by different rules, what you were getting at there?
In the different classic ecological patterns that we look at, for example, latitudinal gradients, the paradigm would be that you see low diversity at the poles and high diversity near the low latitudes. And we see that jump up in the Arctic, and that was surprising. Well, so we've got three different guesses for why, and that'll launch sort of a whole series of efforts to try to resolve which of those hypotheses works. But we think it could be due to riverine input in the Arctic that's different from the Antarctic. It could be from the particular seasonality. We were there in the peak season, though we expected that actually to decrease diversity rather than increase it. And then it could also be to the structure of the Antarctic versus the Arctic water circulation.
Question here.
Alison, you have I'm Robin Williams from ABC in Australia, some water in your cup there. I know it's very difficult to have an average seawater. How many viruses might there be were that seawater? Would it be hundreds of thousands?
Actually, Joshua and I and Matt have done some work on this. So the estimate is that for every single bacterium, there are at least 10 viral particles. And so when we think about open ocean environments, the surface waters, there are somewhere on the order of 1 million bacterial cells per milliliter of water.
So it's a gigantic number.
10 times as many. It's a gigantic number.
Absolutely gigantic number.
And to be honest, our current estimates are likely an underestimate because of some of the technological constraints of trying to make those types of measurements.
And it makes you worried every time you go swimming and you swallow some sea. But what does that mean for the possible change of carbon balance that you've been hinting at?
Yeah, I'll start this, but then I'll let maybe Matt take over. But I think it's really thinking about the roles that when we think about the ocean as sinks and sources of carbon dioxide. And so microbes play really fundamental roles in dictating where that carbon is, if it's going to go back to the atmosphere or perhaps sink to the depths. And so Matt was mentioning the Terra Oceans, and in particular talking about carbon export. And I don't know if you want to talk about the biological pump in that context.
Sure. The idea is that the carbon dioxide comes in as a gas, And then organisms can use that to produce organic carbon or particulate or biomass. And what happens to that biomass is determined by this microbial loop. And so if that microbial biomass is heavy, it might sink, and that would help us because it would sink some of that carbon dioxide out of the ocean and continue the ocean bringing more carbon dioxide in. And so what's been interesting lately is we thought that viruses would short circuit that microbial pump and instead keep all the carbon in surface waters and keep it small and rapidly recycled. And that would be sort of problematic if you were wanting to engineer the oceans as a big carbon sink. But we're instead seeing that viruses best predict carbon flux. And so that's led us to think differently, that that sticky virus material may aggregate and may sink. And so in this case, viruses may indeed be our friend.
Let me just add one more thought to that question too, which is that in terms of this number of 1 million microbes per milliliter, or about 10 million viruses, we also have, this is joint work with Alison and others, we looked at variation in that number. We find significant variation. So just to keep in mind, certain places there'll be about 1 virus per every bacteria. Some there'll be hundreds. So it's important to realize that this variation is quite typical. And so there also could be different drivers, not only in the abundances, but in some of the relative degree to which viruses are lysing material and therefore recycling versus other generating sticky material, which then sinks out. So that's certainly an area of key research at the moment.
Question here? Yeah.
Clive Cookson from the Financial Times. How might human activity, either deliberate to sort of somehow as part of a geoengineering project or inadvertent, how does human activity influence the extent of viral infection of microbes in the oceans?
Yeah, I think that I'm partially in an engineering department as well, so I think about sort of solutions-based use of our knowledge. And there is already talk about developing large-scale bioreactors to grow up viruses, to tweak systems. And before that, there was already the idea of iron seeding the ocean. So many regions of the ocean are very nutrient-limited for iron. And so if you could put a bunch of iron filings in the water, you could cause a bloom of phytoplankton, which could suck down extra carbon dioxide. And so people are thinking about engineering at scale, and it's complicated. But I actually think that the throughput of our methods are approaching what I would argue are ecologically and engineering friendly. And so you could start to imagine in a couple of decades engineered solutions in the oceans.
And sorry, just to follow up. When you say engineered solutions, you mean specifically engineered to help viruses infect bacteria, or are you talking more generally about seeding with iron and so on?
Well, I think both of those. So if you understand the cell-virus interaction well enough, you might be able to adjust with some small nano probe the infection. You could also just add or seed viruses, basically enriching for the interaction that you'd like. And then there's also this idea of basically treating the oceans as a fertilization problem, which I can say has been tried multiple times. The iron seeding has been tried multiple times and been very unsuccessful. But there's a variety of reasons from an ecologist perspective you can imagine that happening. And I think as those communities start talking, as this becomes a critical societal need, reality can set in.
If I add one other element, so as a theorist who studies ecoevolutionary dynamics, one of the other complicating factors, I think, and why we need more fundamental research in this area is that viruses and microbes are continually evolving. And so when we think about solutions here, these are not static players, they're dynamic players. And so one of the things we're very interested in is the degree to which the system and the feedbacks between viruses and hosts, we have to think not just about short-term solutions, so the iron fertilization in one which you get a bloom, but does that lead to long-term change? And so that's one of the issues here. If there is virus modifications of carbon flux, are these short-term responses or long-term responses? And that's really motivates a lot of our research across the board, whether in bringing model organisms to the lab or looking in the field or doing theory of the kind we do.
And maybe just to hit that on the head real quick, this idea of the ecology of phage resistance, so phages are viruses that infect bacteria, and we currently know the ways that a bacterium can be resistant to a virus for maybe 50 or 100 of these interactions. And the numbers earlier were millions per milliliter of seawater. Scale that up to the ocean, you see the problem. And so we're working on high throughput approaches to try to ask that question about phage resistance at an ecologically relevant scale. And sort of until we piece together those kinds of knowledge gaps, it is an engineering nightmare. But I actually see the technologies are there to where we can have great scalability, and we'll see what happens.
Question over here.
Nuala Moran from BioWorld. I wondered if your insights into bacteriophage and how they infect bacteria have any relevance or could inform work in antimicrobial resistance in new forms of treatment for infectious human diseases.
Do you want me to start? You want to start?
Go for it.
So a number of us, I think the general answer is yes, and I think that there's an intersection between studying viruses in aquatic systems, which have been historically the most well-studied and have opportunities for us to understand these questions, and then bringing some of both technologies, insights, and principles of how the systems interact to bear when trying to think about therapeutic phage. And part of the reason is simply, as you likely know, is that phage infect a pathogen. This might eliminate that one pathogen, but are we therefore selecting just for another form of resistance to phage? So part of our work, although it's separate from what I described today, views the phage therapeutic problem through the lens of a system where instead of an aquatic ecosystem, we think of a host-associated ecosystem in which there's immune factors. And so in work with French collaborators at the Institut Pasteur, we evaluated ways in which phage, when combined with an immunocompetent host, could lead to elimination of a multi-drug resistant pathogen that neither the phage nor the innate immune system could take care of by itself. And so this is an area that we think is also ripe for helping these large-scale trials, because certainly the crisis and rise of increasingly multi-drug resistant pathogens combined with a dearth of antibiotics is pointing towards phage as a new either single or most likely combination therapy in concert with other methods.
And Joshua looked at me because he knew my lab's based strongly in the oceans, but we also work quite a bit in humans now. And intentionally, complementarily to Joshua's work, we've focused on this ecology of phage resistance question because we believe that's going to help inform cocktails. Which phages should you put into the treating therapeutic cocktail? And secondarily, we're interested in what I call ecosystem-aware phage therapy. So let's say you're successful and your phage takes out the pathogen. Well, that releases toxins as well as organic matter. How will that impact the other microbes in your body or in the infection? And so we need to understand that sort of ecosystems perspective of the successful phage therapy treatment. And I think it's very nascent, this area. And the stuff that we're doing in the oceans is incredibly informative for it, even though the phages and the other players might be different. The perspectives, the data analytics, the approaches are all readily transferable, and we've demonstrated that extensively in my group.
Oh, I mean, I think just one of the things that I always like to think about is that, a lot of what we know about host-virus interactions when we're talking about bacteria in particular is really based on a very limited number of systems. And so anything we learn about even ocean bacteria provides us with new principles that I think are worth considering in any microbial community.
I think maybe, Alison, you alluded to interactions between viruses and maybe biofilms. Is there a way to harness the power of viruses to stop problematic biofilms from spreading?
Yeah, I think that's a really great question. And I think one of the challenges with biofilms is they're not just cells, it's this extracellular matrix. And that continues to be an issue for viruses because it impedes their mobility from a host cell to another target cell. And And so I think that that's something that people are considering. But then when you think about the types of viruses that we've been talking about where they've been integrated within the genome, if there is a way to trigger that virus to switch to that lytic mode, that would be a mechanism to help alleviate the biofilms by basically initiating those viruses to switch from being more commensal to being more lytic and killing their hosts.
Do you know how they turn on that switch yet?
So I think that that's an area of a lot of interest. I think the paradigm is that host stress is a major driver for a switch. And so when the host cell is stressed, that can initiate that integrated viruses to pop out and become lytic. But I think that, again, I just want to sort of highlight that line of thinking really comes from a limited number of really well-studied systems. And I think that as we're learning more and more about not only the variety of bacteria, but also the viruses that infect them and the ways that they infect them are turning out to perhaps expand upon these earlier notions about just stress is what is causing these things to pop out.
What are the advances in technology that you've been using to get at viruses? Like, what have been the tools that you use?
Yeah.
I'm a theoretician and modeler, and I work with these folks, so I'll defer to them.
I use classic tools. You use a lot of high throughput tools.
So a lot of it's large-scale sequencing and developing approaches to let you get data sets from low input. So when we go to the oceans and collect tens of liters of water, we'll get very, very, very little nucleic acids, DNA, that we can sequence. So we had to develop low-input quantitative approaches. There's a lot of interest in linking viruses to their hosts experimentally. So just because we can survey the ocean and tell you which viruses are there, of course, the most interesting question is who do they infect? And so that's an area of hot topic, and there's probably 9 to a dozen kinds of methods in that space. And then there's approaches that leverage high throughput tools like flow cytometry to allow you to sort based on features. And so we've developed viral tag metagenomics out of that, where you can take a bait culture microbe and pull out 10 to the 7th viruses for that microbe in a 10-minute experiment. So a very high throughput way to capture viruses for a particular host, though it is quite finicky to get set up. But if you're interested, you can do it. That's a smattering of things, but there's a number of others.
And just with that comes quite a lot of data. And so I think the other part when we think about new technologies is also the data analytics side, or how do we make sense and do the inference from these direct observations, which are usually partial, to understand at the end of the day this community-wide, often complicated network of viruses and bacteria. Going back to the comment about 10 to the 7th, which is 10 million viruses per milliliter and a million or so bacteria, and there's a lot of variation. Not all of those infect each other, right? So there's, relatively speaking, specialized pairs which form in total a network. And so in work that we're trying to do from the theory side is how do we take this omics data, how do we take observations of changes of these different types and then infer what would be the most plausible set of interactions that could make sense of all these observations from omics level data. And so I think that's going to be an important partnership in the next few years, and one that we're recently funded on, Matt and I, with Steve Wilhelm, a colleague and collaborator of Alison Buchan at UT Knoxville, to work on.
And on the analytics front, I mean, I think that a nice thing is we've, as a community, developed a platform called iVirus, which takes these viral ecogenomic analyses and places them in a cyber infrastructure funded by the NSF that democratizes that capability. So for a long time, it was if you didn't have a programmer in your lab, you couldn't see the viruses, even though they're right there in a lot of these data sets. And so now a lot more people can see viruses in these data sets with iVirus.
Question up here.
I remember 10 or 15 years ago when Craig Venter went out sailing and analyzed seawater and extracted the DNA, and he found a gigantic number of bacteria. I don't remember him talking about viruses then. Was that viral DNA neglected, or was it just not reported by journalists like me?
Actually, that's a great spin-off of the iVirus thing. Those data were in those data sets, but they didn't really specifically sample the viral fraction, so they would have underreported it. And then because the databases weren't there, they couldn't really see a lot of the viruses in the data they did collect. Though now you could see a lot of information from that. Of course, the data set's a smaller scale than what we can generate now. But
So do you think that most bacteria in seawater are infected with viruses, or are some of them virus-free?
we're all ready to jump in.
Yeah. Alison, do you want to start? Or?
I would venture to guess that all are susceptible to viral infection, whether at any given time they have a active viral infection, however you want to define that. I think that they don't necessarily always have to have a virus associated with them. And so I think one of the things that we're generally interested is understanding features like, well, given the immense diversity of bacteria, given the immense diversity of viruses, how do the right virus-bacteria pairs, how do they find each other to interact, right? And then also thinking about these specific type of viruses where they can lie sort of quiet within the host genome. What are the kinds of triggers that cause them to pop out and adversely affect at least that individual host?
And I think, to give you some numbers, so if you do tracer experiments, about 1/3 of cells are infected per day, and there's variation around that number, of course, but that's a kind of good rule of thumb number. And then if you sequence genomes from the ocean, 50% to 2/3 would have a virus in their genome, these temperate viruses as a prophage. And so you both have lytic infection and you have viruses inside genomes that often can regulate the metabolism of that cell, and also, of course, are sort of waiting to become lytic and kill that cell depending on conditions.
And this goes beyond also the ocean. So in separate work, we've looked at hot springs and other environments where we found, we had a paper that was entitled "A Virus or More in Nearly Every Cell." So we're looking at single amplified genomes and finding that there were at least one, if not more than one virus. And in some of, I know your work, you're finding associations between even different genetic modes of viral infections in the same cell, whether RNA and DNA. That also motivates some of our theoretical work, which is, do we think that certain environments will favor or predispose viruses to adopt this lytic mode or this temperate mode? That could be varying by season. So for example, in low productive, low density situations, there's evidence that that would lead to or associated with a higher fraction of this temperate mode. And then we find more infected cells, so visibly infected cells in periods of high density. And that's some work from a few years ago, and that also begins to ask questions of if viruses can do both, because when they integrate, they can come out. So there is this choice. What are the ecological conditions that favor it? And that has to do with, at some point, the virus's fitness is entangled and associated with the cell. If the cell divides and does well and the virus is inside, it's doing well. But when the cell may not be doing so well, it may be time to go and get a different host. So that question of infections, infected cells, as well as this temperate mode, and the modes to look at them are quite different.
Speakers had a video I wanted to make sure that we could play. So Darren, could we cue that up while we have just a few more minutes?
Marine microbial communities are uniquely responsible for carrying out processes that are essential for all of Earth's biogeochemical cycles. The function of a microbial community is in large part dictated by the composition, who's there, and how many of each. This means that in microbial communities, bacteria compete with one another for resources. In this fight, some bacteria can produce antibiotics and use them against other types of bacteria. This kind of interaction has been known for some time. But there's another fight strategy that scientists are just now considering. Bacteria might use the same viruses that infect them as weapons. In this context, defense becomes a necessity, and we recently discovered another form of resistance. Dying microbes can produce a new virus that then go to attack the the original invader. This type of competitive interaction is important for stabilizing microbial populations in marine systems. And since many processes involved in climate change are microbially mediated, having a better understanding of the relationships between bacteria and their viruses could help us design better strategies for climate change mitigation in the future.
It's very well done. Okay. Do we have any other questions today? Do we have any other comments before we end? Okay. All right. Thank you so much to all of our speakers. That was a great briefing. Appreciate your time.
Thank you.